Using an artificial intelligence algorithm, researchers at MIT and McMaster University have identified a new antibiotic that can kill a type of bacteria that is responsible for many drug-resistant infections. If developed for use in patients, the drug could help to combat Acinetobacter baumannii, a species of bacteria that is often found in hospitals and can lead to pneumonia, meningitis, and other serious infections. The microbe is also a leading cause of infections in wounded soldiers in Iraq and Afghanistan. Acinetobacter can survive on hospital doorknobs and equipment for long periods of time, and it can take up antibiotic-resistant genes from its environment. It is really common now to find Abaucin isolates that are resistant to nearly every antibiotic. The researchers identified the new drug from a library of nearly 7,000 potential drug compounds using a machine learning model that they trained to evaluate whether a chemical compound will inhibit the growth of Abaucin. This research appears in the journal Nature Chemical Biology. Over the past several decades, many pathogenic bacteria have become increasingly resistant to existing antibiotics, while very few new antibiotics have been developed. Several years ago, MIT researchers set out to combat this growing problem by using machine learning, a type of artificial intelligence that can learn to recognize patterns in vast amounts of data. They hoped this approach could be used to identify new antibiotics whose chemical structures are different from any existing drugs. In their initial demonstration, the researchers trained a machine learning algorithm to identify chemical structures that could inhibit growth of E. coli. In a screen of more than 100 million compounds, that algorithm yielded a molecule that the researchers called Abaucin after the fictional artificial intelligence system from 2001, A Space Odyssey. This molecule, they showed, could kill not only E. coli but several other bacterial species that are resistant to treatment. The researchers says after that paper, when they showed that these machine learning approaches can work well for complex antibiotic discovery tasks, they turned their attention to what they perceived to be public enemy number one for multi-drug resistant bacterial infections, which is Acinetobacter. To obtain training data for their computational model, the researchers first exposed Abaucin grown in a lab dish to about 7,500 different chemical compounds to see which ones could inhibit growth of the microbe. Then they fed the structure of each molecule into the model. They also told the model whether each structure could inhibit bacterial growth or not. This allowed the algorithm to learn chemical features associated with growth inhibition. Once the model was trained, the researchers used it to analyze a set of 6,680 compounds it had not seen before, which came from the drug repurposing hub at the Broad Institute. This analysis, which took less than 2 hours, yielded a few hundred top hits. Of these, the researchers chose 240 to test experimentally in the lab, focusing on compounds with structures that were different from those of existing antibiotics or molecules from the training data. Those tests yielded 9 antibiotics, including one that was very potent. This compound, which was originally explored as a potential diabetes drug, turned out to be extremely effective at killing Abaucin but had no effect on other species of bacteria. This narrow spectrum killing ability is a desirable feature for antibiotics because it minimizes the risk of bacteria rapidly spreading resistance against the drug. Another advantage is that the drug would likely spare the beneficial bacteria that live in the human gut and help to suppress opportunistic infections. In studies in mice, the researchers showed that the drug, which they named Abaucin, could treat wound infections caused by Abaucin. They also showed in lab tests that it works against a variety of drug-resistant Abaucin strains isolated from human patients. Further experiments revealed that the drug kills cells by interfering with a process known as lipoprotein trafficking, which cells use to transport proteins from the interior of the cell to the cell envelope. Specifically, the drug appears to inhibit LOIE, a protein involved in this process. All gram-negative bacteria express this enzyme. So the researchers were surprised to find that Abaucin is so selective in targeting Abaucin. They hypothesized that slight differences in how Abaucin performs this task might account for the drug's selectivity. The researchers also plan to use their modeling approach to identify potential antibiotics for other types of drug-resistant infections.